Hello, and welcome to a video about protein structure and function. In this video, what I'll be talking about is the four different levels of protein structure and how those structures relate to its function with the two different types of protein. So there's four levels of protein structure: primary, secondary, tertiary, quaternary. Primary is your sequence of amino acids in a chain. Your secondary is beta pleated sheet to alpha helix. Your tertiary is how this chain interacts with the beta pleated and the alpha helices, how they interact with each other, and quaternary structure, which is more than one protein consisting of more than one amino acid chain. So your primary structure is your amino acid chain. It's the order of the amino acids in that chain. So DNA is transcribed and translated. For more information on this specific part, so how we go from DNA to our amino acid chain, watch the video of DNA to protein that I've made a couple of weeks ago. This brings us to our secondary structure. Now, secondary structure is one of two things. It can either be an alpha helix or beta pleated sheet. And whether it's an alpha helix or beta pleated sheet will be determined by the amino acids in the primary structure, so the chain of amino acids. Depending on where the amino acids are that can form hydrogen bonds will determine whether it's an alpha helix or beta pleated sheet. So for beta pleated sheet, it's hydrogen bonds between the backbone of every fourth amino acid. And the R groups stick out in this. Now remember, the R group is a functional group of the amino acid. And that's what sticks out in an alpha helix. Whereas in the beta pleated sheet, you get hydrogen bonds between backbones of adjacent strands. And the R groups stick out in alternating fashion, so they go up and down, up and down, up and down. And that will obviously make a difference to the way it interacts in the tertiary structure, which is what we're about to talk about. But basically, your secondary structure is either alpha helix or beta pleated sheet. This brings us to our tertiary structure. Now, you need to remember that proteins only do what they're meant to do by being a very specific shape. So this shape is determined by how it's folded. So from our primary structure, we have the amino acid chains, which determines alpha helixes and beta pleated sheets in our secondary structure. Now, the R groups of those amino acids will also further interact, and this will lead to the protein folding, to the amino acid chain folding, and becoming a tertiary structure. So, tertiary structure are interactions between the side chains of R groups, and with the way it folds, it's determined by a couple of different things. So you can form peptide bonds, disulfide bonds, ionic bonds, hydrogen bond, and hydrophobic interactions. So if the amino acids have two sulfurs, therefore you form a disulfide bridge, which will bring those two parts of the amino acid chain together, closer together. Ionic bonds between a positive amino acid and a negative amino acid, R group, will bring that part of the chain closer together. And hydrogen bonds between a hot hydrogen and oxygen or hydrogen and nitrogen of your amino acid will again pull that chain close together. So it's the way it's folded together which gives it its function as well. So to recap what we've said so far, we've got the primary structure, which is the amino acids sequence. We've got second structure, which is alpha helix or beta pleated sheet, depending on where the R groups are that can form the hydrogen bonds. And we've got tertiary structure, which is the folding of that amino acid chain. This then brings us to our last structure, which is a quaternary structure. Now, the quaternary structure is based on if you've got more than one amino acid chain and a protein. It's how these chains will interact. And the interactions are the same as a tertiary structure, but they're between different amino acid chains. So it's how it folded, how it makes that shape, and that's where proteins will get their function from. Remember, shape is what determines what the protein does. So to fully recap, what we said is your primary structure is your amino acid chain. Your secondary structure is alpha helix or beta pleated sheet. Your tertiary structure is how does alpha helix and beta pleated sheets all fold together, depending on the interaction between your R groups. And your quaternary structure is how more than one amino acid chain will fold together and shape together using the same interactions as a tertiary structures to determine the overall shape of the protein. This now brings us to different types of proteins. So the first protein we're going to talk about is globular proteins. Now, globular proteins are given the name globular because they look spherical, roughly in shape. They're compact, they're water soluble, and that means they will have metabolic functions. So, most enzymes, for example, are globular proteins. And with globular proteins, the hydrophilic R groups are kept on the outside of the protein, which allows it to be soluble in water, which is different from fibrous proteins, but we'll talk about that in a little bit. And this solubility is what makes it so important for its functions. So they regulate many processes that are necessary for life. The main example of a globular protein I want to discuss today is insulin. Now, insulin is the hormone that's involved in regulation of blood glucose concentration, and because it's transported in the bloodstream, it has to be soluble. That's where the importance of globular proteins being soluble comes in. They have to fit, because the hormone has to fit into a certain specific receptor, so the shape is really important. So the precise shape, it's soluble, it's small, it's globular. So insulin is an example of a globular protein. Conjugating proteins are also a type of protein. Now, conjugated proteins are basically globular proteins that contain a non-protein component called a prosthetic group, which means a non-biological group. This could be lipids, it could be carbohydrates, so forming lipoproteins or glycoproteins. It could be, in the case of hemoglobin, as is in the diagram here, it could be the heme group. Now, the heme group is just iron 2 plus. Okay, so that is a conjugated protein because it's a protein that's associated with a non-biological group, a prosthetic group, if you like. Catalase is an example of an enzyme that is also a conjugated protein. So these proteins have functions based on what their prosthetic group does. So, for example, hemoglobin, the fact that you've got iron 2 plus that allows the hemoglobin to pick up oxygen to take around the body. Without the iron 2 plus, it wouldn't be able to do it. So our last protein type is fibrous proteins. Fibrous proteins are there for structure. So they have long, insoluble molecules. They have a majority of amino acids with hydrophobic R groups. These R groups on the outside, so it's not soluble in water. And they tend to make strong, long molecules which are not folded into complex three-dimensional shapes like globular proteins. So examples of fibrous proteins are things like keratin, elastin, collagen, which are all strong fiber-like proteins which are there to provide structural support and strength. These are tensilely really strong because of the interaction between the layers of the fibrous protein. So to recap the difference between a fibrous and globular protein are compact protein structures, fibrous extended. Globular is soluble in water, fibrous is insoluble. Globular has secondary structure, which is complex through a mix of alpha helixes, beta pleated sheets, and loop structures, whereas fibrous would generally only have one simple secondary structure. Quaternary structure is held together by non-covalent forces in globular proteins, whereas in fibrous proteins, quaternary structure is usually held together by covalent bridges. And fibrous is all to do with structure in the body, whereas globular proteins all to do with functions in the body. I hope this helped. Please let me know if you have any questions, either by email or in the comments below. Thank you.